Hello everyone, welcome to Bio Academy. Today we will discuss about a very simple thing which is very important as well. We will discuss in a detailed manner. You must have known about DNA, which is the genetic material. You can see here a helical structure. In the left one you can see the terms sugar, base pairs, phosphate, hydrogen bonds, etc. You can see the structure. The complex structure can confuse you guys, but no need to afraid. We will discuss in a simple way and detailed information you will get in this video. What is a DNA? Do you know? So let's begin. It can be denoted as deoxyribonucleic acid. D stands for deoxyribose, N stands for nucleic and A stands for acid. Okay? After that, there are two types of nucleic acid, DNA and RNA, which can denoted as deoxyribonucleic acid and ribonucleic acid. So the monomeric unit of nucleic acid is nucleotides. We can say nucleic acid is polynucleotide sequence. So what is a nucleotide? We can signify it as phosphate ester of nucleosides. So if we simplify it, then we can say that nucleotides composed of three things, that is nitrogenous bases, sugar, phosphoric acid. The sugar making nucleic acids are of 5 carbon, which we can say ribose, deoxyribose, which is lacking a hydroxyl group or OH group. Okay. Next, what is a nucleoside? It composed of sugar and nitrogenous bases. Now, what is nitrogenous base? It is a heterocyclic planar of aromatic bases, which is two types, purines and pyrimidines. What is that? No need to worry. It is very simple. We will learn it in our next slide. So, purines. Heterocyclic aromatic compound means two cycle or two rings. You can see in the diagrams. You have to remember the numbering of the structure so that it will help you to memorize the naming of the purine types. In the first, 7th and 9th position, N or the nitrogen is present. Two types of purine are there, which is adenine and guanine. Two common purines are adenine and guanine. So the first one is adenine, which can be named as 6-aminopurine, which is soluble in water. It pairs with thymine by double bond. From the name, we can see that the amino group is present in the 6th position. In this structure, you can see that position of amine. Second one is guanine. The chemical name is 6-oxy-2-aminopurine, which is insoluble in water. In the C2 position, amine group, and at C6 position, a carboxyl group is present. You can identify it in the structure. Pyrimidines is simple aromatic ring. Two major pyrimidine in DNA are thymine and cytosine, and two major pyrimidine in RNA is cytosine and uracil. In RNA, T is replaced by uracil. You can see the general structure. See the numbering as well, which will help to understand the structural name. Now, thymine, 5-methyl-2,4-dioxypyrimidine is its chemical name, in which in C5 position methyl group is there, and C2 and C4 position the carbonyl group. You can see the structure in right hand side. Next is cytosine. Its chemical name is 2-oxy-4-aminopyrimidine, indicating the carbonyl group in C2 position and amino group at C4 position. Next is uracil. Its chemical name is 2,4-dioxypyrimidine, which is similar to thymine except the methyl group at C5 position of thymine. You can compare both the structure and exception. You can understand the whole in a simple way. The nitrogenous base and sugar forms nucleosides. In the nucleoside, if a phosphate group is added, forms nucleotide, and if a number of nucleotide aggregate forms polynucleotides, which is also called as nucleic acid. Now, after the nitrogenous base, the other part of the nucleic acid is sugar, which contain 5 carbons or called ribose sugar. 5 carbon containing ribose sugar is present in RNA and 5 carbon containing ribose without a hydroxyl group or OH group at 2' position of sugar, which is replaced by hydrogen and called deoxyribose, is present in DNA. So you can see in the picture and can compare. The difference you can identify in the 2' position. When we study nucleotide H whole and H we have already named 1, 2, 3 and 4 in the nitrogenous bases. So here the name for sugar is mentioned as 1', 2', etc. Bonds or connection we can identify in a nucleotide and nucleic acid which links between the nitrogenous base, phosphate, sugar, etc. One type of bond is phosphodiester bond, which is a covalent bond. You can see the bond in a nucleotide structure in next slide. Now glycosidic bond, which is a connection between sugar and nitrogenous base. You can see in the diagram, which is a glycosidic bond. Now polynucleotide. If more nucleotide condense forms polynucleotide. Here you can see the phosphodiester bond linking the nucleotides. The bond formed between the 3' OH of one nucleotide with the 3' phosphate of other nucleotide. 5' phosphate of other nucleotide. Now the polynucleotide can form nucleic acid, which is of two types, DNA and RNA. The double helical structure of DNA described by Watson and Crick in 1953, in which two individual strands align in anti-parallel fashion. Now one strand in 5' to 3' manner, whereas the other in 3' to 5' direction. Three forms of DNA we generally study. They are A, B and Z DNA. Before coming to detail of A, B and Z DNA, we can first describe about Watson and Crick model. So it will help for better understanding. So Watson and Crick described in their model that DNA is right-handed helix in which a long polynucleotide strand coiled around a central axis anti-parallelly means facing opposite to each other, or we can say that the direction of two strands will be 5' to 3' and 3' to 5' orientation, and the two anti-parallel strands interact by hydrogen bonding between the complementary base pairs, in which adenine pairs with thymine by 2 hydrogen bonds and guanine pairs with cytosine by 3 hydrogen bonds. The diameter of helix is 20 angstrom and helix rise per base pair, or we can say that the distance between two base pair in the strand is 3.4 angstrom. In a helical turn, 10.5 base pair present and interaction forms major and minor groove. In this diagram, you can see the picture of bDNA. The diameter is 20 angstrom. You can see the base pair per turn, major and minor groove, axial rise, etc. And when we study the different types of DNA, you can compare the data. Now let's discuss about the types of DNA. First is aDNA. It is also a right-handed helix with 10.7 mean base pair per turn, 23 angstrom diameter, 2.3 angstrom rise per base pair. Major groove is narrow and deep, whereas minor groove is wide and shallow. Sugar pucker is C3 endo. Pentose sugar is non-planar and the non-planarity is termed as puckering. Now in bDNA, the helix is right-handed, 10.4 base pair per turn. Pitch per turn is 33.2 angstrom, 20 angstrom diameter and 3.32 angstrom rise per base pair. Now the major groove here is wide and deep and the minor groove is narrow and deep and puckering is C2 endo. Next, ZDNA is left-handed. The diameter is 18 angstrom. The mean base pair per turn is 12. The pitch per turn helix is 45.6 angstrom. The rise per base pair along helix is 3.8 angstrom. Here the major groove is flat and minor is narrow and deep. The puckering here in cytosine is C2 endo and C3 endo in guanine. We can discuss the comparison between the three DNA types so that it will be helpful for us to remember the major important data. You can see here the three DNA A, B and Z. First point, if we take sense of the helix, then A and B DNA are right-handed helix, whereas the Z DNA is left-handed. The diameter of A DNA is 23 angstrom, B DNA it is 20 angstrom, and you can see in the Z DNA it is 18 angstrom. Now the mean base pair per turn, or you can say how many base pair is present in one turn of DNA, and that is 10.7, 10.4 and 12 in A, B and Z DNA respectively. Next is rise base pair per helix, means the distance between two base pair in DNA, and that is 2.3 angstrom in A DNA, 3.32 angstrom between the base pairs in B DNA and 3.8 angstrom in Z DNA. Now the pitch per turn of helix is 24.6 angstrom in A DNA, whereas 33.2 angstrom and 45.6 angstrom in B and Z DNA respectively. We have individually discussed about the topics, but this is a comparison in between, so that will help in better understanding. The major groove you can see narrow and deep in A DNA and wide and deep in B DNA and flat in case of Z DNA. And the minor groove you can see wide and shallow in case of A DNA and narrow and deep in B and Z DNA. So this is the comparison, and if there is any confusion in the character like the pitch for helix, axial rise or rise per helix, diameter, then you can refer the diagram in previous slide. and can compare the value of different DNA in that picture. These are the references you can follow for more information. That's all for today. Thanks for watching.